So hello everyone, good morning. Thank you all for joining this essential concepts and techniques of the GIBA. So as I've been sending around some emails to all the participants, so we'll be having 3 different morning sessions throughout this week around each one of the GIBA, so one of each of the axes of our network, so the gut, immune, and brain. As you may have noticed, the cameras and mics have been disabled. This is because we expect quite a large number of participants, so we're trying to minimize any potential technical issues. But please, I do encourage you to use the chat, especially for the Q&A, for asking any questions to the speakers. Please contribute through the chat function. I just started the recording, so all these lectures will be recorded and made available to the whole network. So if you didn't manage to catch something or wanted to review again the lectures, you'll be able to do so at a later point. And in terms of the structure, so we're going to have, for each of the sessions, we're going to have 2 speakers. Each one will give a talk of about 40 minutes, and then we'll have a chance of about 15 to 20 minutes of Q&A. So please use the chat for that. So for today, we're going to start with John McClellan, and then we're going to be followed by Leslie Hoyles. So John is a professor of gastroenterology and nutrition at University of Manchester, and he also works as an honorary consultant gastroenterologist at the Salford Royal Hospital. And his current research interests are around the gastrointestinal physiology in health and disease, with a particular focus on the interactions between nutrients and the gut epithelium, gut inflammation, gut hormones, and most importantly, the gut-to-brain signaling. So John is going to give us a bit of an overview of gut physiology, structure, and function, and Leslie is then going to follow with a specific talk around the gut microbiome. So John, whenever you're ready, feel free to get started. Thank you all.
Yeah, thanks, Alex. I think you can see my slide already. So
yes,
that's good.
All good.
Yeah, so brilliant. So thanks for that introduction. So I was very pleased to see the GIBA network set up. Actually, I was part of the BBSRC working group that led to this. So it's really nice to see it come to fruition and being so active now. So thanks for that. So just to tell you what I'm going to do today, this is really very introductory and quite basic talk, mainly probably for people who are not very versed with the gastrointestinal tract. It's probably around BSc level is what I was advised. So if you find what I'm saying is a bit too basic for you, then feel free to drop out for a while. I'm going to give you a bit of a top to bottom tour, literally through the gut and its functions. Not really going to say much about immunology, neuroscience, and microbiota, because these are going to get covered in proper detail in other talks. So I've really kept largely away from those with a few exceptions. And also, I'm quite, as Alex said, I'm quite interested in gut hormones, gut endocrine cells. So I'm going to give a little bit of extra stuff and some slightly old research that we did now, but something that I think I'd like to see come back into the fold. So back to basics. The gut's interesting. It's got 2 quite conflicting roles. Clearly, the reason we have a gut is for digestion and absorption, so to support our nutrition by getting food and water into our bodies. But it's also got a barrier function. It's got to keep the bacteria, the toxins, and all the bad stuff out. And the gut's really very carefully designed and adapted to achieve these conflicting roles. And of course, you've got to absorb nutrients with maximal efficiency. Clearly, our eating and digestion and absorption are essential, highly evolved and closely regulated. But having safe, easy to get diet is really quite a recent thing. It's in biological terms, it's quite recent that we've been able to cook and process food and preserve things. And our biology is really designed for when things were much harder to get. And so in a sense, it makes more sense to eat as much as you can when it's available to stay alive. Rather, ideas of obesity and so forth being due to a problem of the gut are just not true, because of course we're designed to maximize digestion and absorption. So as Alex said, I'm a gastroenterologist. I'm not really going to talk about diseases much, but in my clinics, we'll see people with all sorts of problems with the gut, so indigestion, heartburn, vomiting, abdominal pain, bowel problems, bleeding, weight loss, and many of these things are related to immunological and inflammatory problems in the gut. But also in this situation, we've got to consider the role of the gut in appetite regulation, and how being overweight and obese can follow from nutrient excess. which of course underpins the surge in recent treatments such as the GLP-1 and GIP analog drugs that are really changing obesity management. But also we've got to think about the role of the gut in healthy, lifelong nutrition, not just in disease terms. As gastroenterologists, sometimes we try to simplify things a little bit about when there are problems with the gut. Was it a problem with the plumbing, so is something narrowed or blocked? Or is it a problem with the wiring? Is the problem with the gut nervous system and its controls? But increasingly, we've got to think about how the immune system interacts with all these gastrointestinal functions and dysfunctions. And the other thing for context is just to put the brain-gut axis in there. So there's close bidirectional connections between the gut and the brain, which affect each other. And things, psychological factors and stress can influence this, as can diet, as can the microbiome, as you'll hear later, just the normal microbiome, but also if there's gut infections or antibiotics changing the microbiome. Really interesting paper last week from Sweden suggesting that antibiotics can change the gut microbiome for up to 8 years from a single course, which is quite a thing. And of course, our genomes and the microbiome genomes all interact with the gut to brain axis, which is probably quite all important in appetite regulation and dysregulation. But also we increasingly recognise disorders of gut-brain interaction. So things like irritable bowel syndrome, for example, we now consider to be a disorder of gut-to-brain signalling. And of course, then for the purpose of GIBA, we've got to think about how the immune system interacts with all this. So that was just some background context about the gut. And now I'm just going to give you, as for this kind of tour of the gut from top to bottom, just to let you know about the different structures and functions of the gut. Most of this is obvious and you know this, but just as a refresher. So of course the mouth, mastication, chewing, getting things into small boluses that are ready for swallowing by the oropharynx, and then things going to the esophagus, where peristalsis transports boluses of food down to the stomach. The most common disorder here is gastroesophageal reflux, where gastric contents and acid wash back into the esophagus and cause inflammation. So that's something called esophagitis. And that can progress to a condition called Barrett's esophagus, which is the precursor for esophageal cancer, which is actually getting more common in the West, probably due to diet and obesity being large components of that. So this is what as gastroenterologists we see using endoscopy cameras. So on the left there's a normal esophagus, and on the right quite an inflamed esophagus with esophagitis that will cause heartburn and indigestion, and it's clearly an inflammatory response to acid and enzymes and things that aren't supposed to be in the esophagus. Over time it can progress to this thing called Barrett's esophagus, which is the lining of the stomach effectively replacing the lower lining of the esophagus. And about 1% per year of those will turn into this esophageal adenocarcinoma, which is one of the least survivable cancers and has a very, very poor prognosis. So next, the stomach. So the role of the stomach is to mix the ingested food and liquids into chyme, so a liquid that goes into the small intestine for digestion and absorption. The main other thing the stomach does is secrete acid, which is probably a bit redundant now. I think that's because we've got such clean food and water, so you don't acid to sterilize things is no longer strictly required, but it's still there. That's mainly controlled by the vagus nerves and a hormone called gastrin. And of course, acid secretion can be blocked by using drugs called proton pump inhibitors, which are very frequently used for things like acid reflux. The stomach also makes something called intrinsic factor, which binds to vitamin B12 to allow it to be absorbed in the small intestine. And in terms of inflammation and immunology, the key thing that happens in the stomach is inflammation called gastritis, the commonest cause of which is a bacteria called Helicobacter pylori, the discovery of which led to the Nobel Prize for Professor Marshall a few years ago. But gastritis is the precursor for gastric and duodenal ulcers and also for gastric cancers. So very, again, an inflammatory process leading to pathology. This is how gastric acid is secreted. So these cells called the parietal cells line the stomach and produce acid, but they do this in response to histamine, which is produced by these other cells that respond to the hormone I mentioned called gastrin, and also acetylcholine from the vagus nerves stimulate this as well. But also mechanical stretch and the presence of amino acids in the stomach stimulate the production of gastrin. And then it's a nice physiological regulatory loop, because as the acid falls, then another cell type called the D cell produces somatostatin, which locally inhibits that. So you've got a classical kind of physiological feedback pathway inhibiting acid secretion. Helicobacter can stimulate additional acid secretion, and that's what drives the pathology. So onto the small intestine, which is responsible for digestion and absorption. So there's 3 parts to the small intestine. The first part is called the duodenum. That's because it's about 12 inches long, so it's from the Latin for 12. The jejunum, the middle bit, and then the ileum. And then these are connected to the biliary tree and to the pancreas. And this is where the bulk of digestion and absorption of nutrients takes place. If there's a very large surface area to the small intestine, if it's all folded out and flattened, it's said to be about the same size as a standard tennis court. So that's how much gut surface area you've got for digestion and absorption. And that's partly because it's folded into these rings called plici. But then also, if you go down to that, these finger-like projections called villi also massively increase the surface area of the small intestine. So those are the villi lined by absorptive enterocytes, but some other cell types that I'll tell you about shortly. So all this is really there to produce a very, very large surface area so that we can maximize digestion and absorption of precious nutrients. The pathological example where that goes wrong is celiac disease. So that's an immune reaction to gluten, mainly from wheat in foods, and you get flattening of the villi and loss of that surface area, which therefore leads to diarrhea and reduced absorption of key nutrients. But if people stop eating gluten, then that almost always goes completely back to normal. So back to the epithelial cells. So most of the epithelial cells are absorptive enterocytes, which produce various enzymes and transporters to facilitate digestion and absorption. This is a villus, and at the base of the villi there are crypts, and the stem cells are in the crypts, and you get this migration of cells up the villus until they're shed by apoptosis at the tip of the villus. That actually takes about 5 to 7 days. So the lining of the intestine is massively turning over and renewing itself about every week. So there's the absorptive enterocytes, but then there's also other cells, so goblet cells are the cells that produce mucus. So mucus is partly a barrier to on the intestinal surface, but also a lubricant to help things to move through the gut. In the base of the crypt, there's cells called Paneth cells, which produce various defense molecules to protect the gut from infection. And then endocrine cells, which I'll tell you a little bit more about later, my favorite cell type, and then the stem cells. But of course, there's immune cells and immune signaling also taking place in the epithelium, but I'm going to leave colleagues to tell you more about that. That's the inside lining of the gut, the mucosa and the epithelium, but of course there's various other layers and on the outside muscle layers to help contract and push things through the gut. So quite a complex structure, but I think it's remarkable that the epithelium renews itself so rapidly. So in terms of transport, so these are the absorptive enterocytes, and they have their own microvilli on the surfaces, which increases the surface area even more. And there's various forms of transport. Most of it is transcellular through the cells, through a variety of transport mechanisms and transporters. There's also some passive diffusion into the gut. And the key other thing is the tight junctions that are between the absorptive enterocytes, a variety of molecules that cause a tight seal to stop things moving through the paracellular pathway. But of course, when there's inflammation, you've probably heard of the idea of a leaky gut, then that's when this paracellular pathway has become more permeable and allows things to go through it, which it shouldn't do otherwise. There's also some endocytosis as well. And so the concept of how leaky the gut is that of intestinal permeability, which is very much under immune and neural regulation. So in terms of digestion and absorption, there's a whole host of enzymes. So every set of nutrients has got its set of enzymes to break it down to its components. So on the anthracite brush border, there's a variety of disaccharidases that chop up small sugars into single sugars for transport. There's peptidases that break down protein structures into amino acids and dipeptides for absorption. Then the bulk of the digestive enzymes come from the pancreas. So amylase, digesting starch, lipase for fats, and trypsin and other proteases breaking down proteins in the diet. Most of the pancreatic enzymes are actually secreted as inert precursors and only activated in the lumen of the gut, which makes sense because otherwise if they were activated inside the pancreas, that would self-digest, which is what actually happens in acute pancreatitis. The other thing then is bile. So bile is produced in the liver and stored in the gallbladder and released in response to food via this hormone CCK, cholecystokinin, which is released mainly in response to fats in the duodenum. And bile is necessary for emulsifying fat for digestion and absorption. Fat's probably the hardest part of the food to digest and absorb, which I guess when you're doing your washing up, it's the greasy things that don't come off very easily, and you need detergents to get those off your pots and pans, and bile is really that detergent within the gut. And then there's a variety of transport mechanisms, as I mentioned before, on the enterocytes. They've got various transporters for monosaccharides, amino acids, dipeptides, fats, metals like iron and copper, ions and water, inevitably, various vitamin transporters, and then bile salts are reabsorbed in the last part of the small intestine and recirculated back to the liver. And of course, some of these can function as drug transporters also. So moving towards the end of the gut, the large intestine, so things move quite quickly through the small intestine. Probably on average, it's about 60 minutes for things to get from the stomach to the end of the small intestine. But things in the colon move more slowly, over hours, and for some people, many days. We have some people who only move the bowels about once a week or so, and that's normal for them. But the main job of the colon is water reabsorption. It's where the bulk of the microbiome resides and ferments fiber, salvages energy, and has various immune interactions, as you'll hear in subsequent parts of this series of talks. And then right down at the bottom, we've got the anus and rectum, which are simply responsible for defecation. But we see a lot of clinical issues in there with regard to constipation and things like fecal incontinence, unfortunately. So just going back to the gut-brain axis in all of this, because we really only have conscious awareness of what's going on in our mouth and pharynx and in the anus and rectum. We're not really aware of what's going on in most of the gut most of the time. So most of that is subconscious in physiological terms. So this is a little diagram to help just look at the different parts of the gut and what's happening. So a lot of the gut hormones are secreted in the proximal gut in response to nutrients to stimulate bile and pancreatic secretion. And all this digestion takes place in the small intestine with iron and calcium mainly absorbed in the duodenum, sugars and amino acids mainly in the 2 meters on average that we have of jejunum. And then as we go down the ileum, most of the sugars and amino acids have usually been absorbed already, but fat-soluble vitamins are being absorbed still in the ileum, and then vitamin B12 and bile salts absorbed in the last part of this. And the colon, we got on average about 1 meter, but some people can have almost 2 meters of colon, and that's responsible for water salvage mainly. And you've got this increasing number of goblet cells as you go along the gut, and interesting, the villi start taller in the proximal small bowel and get shorter as it goes along. So I found that quite a useful little schematic for how different parts of the gut work. Because we've got quite a large amount of small intestine, you can lose about half of it surgically, for example, and still be able to absorb normal food and function without part of the gut. But we do have some patients who lose an excess amount of gut and have got so-called intestinal failure, and they need long-term intravenous nutrition. So the other concept to have in here is gastrointestinal motility. So things have got to move along the gut, both ingested material and the secretions, and of course all the parts of the gut have got their own motility that move things from the top to the bottom. The small bowel is obviously the longest part where most of this happens. One thing that people don't appreciate is the gut actually makes about 10 litres of its own secretions every day. So there's an awful lot of fluid being secreted in terms of saliva, gastric juices, bile, pancreatic juices. But then only on average, only about 100 to 200 millilitres of water are present in feces in 24 hours. So the vast majority of that ingested and secreted fluid is absorbed within the gut. So things that move along the contractile forces of peristalsis, which move things in a steady top to bottom direction, except during vomiting, of course, when you get reverse peristalsis, but that's a pathological thing. So peristalsis involuntary, it's under intrinsic control. You can't consciously change it, or that is externally modulated by parasympathetic and sympathetic inputs. Psychological and pharmacological factors can influence influence peristalsis, as can the microbiome. And so this is a food bolus being pushed down in the intestine with acetylcholine responsible for contraction above the food bolus, and then BIP and nitrous oxide responsible for relaxation distal to it. So you get this pushing of the bolus along the gut. So for the last part of my talk, I just wanted to go back to enteroendocrine cells, which I think are a little neglected in research terms, and I think are probably more important in terms of their interactions with the immune system than has been generally appreciated. Curiously, that the gut epithelium is actually the largest endocrine and sensory organ in the body. So this would be an endocrine cell with these little microvilli that sense nutrients in the gut lumen and then signal across the cell to release the secretory granules of hormones that have their relevant effects. Only about 1% of the epithelial cells are endocrine, but of course, because the gut is so huge, that adds up to quite a large volume of endocrine material. So nutrient sensing. And the interesting thing, they express various chemosensory receptors, so fatty acid receptors, but also things like sweet taste and bitter taste receptors that you find in the mouth have been found on the endocrine cells. And they also express some immunological receptors, I understand, as well. So this is the purpose of endocrine cells, to encode transepithelial signaling, so the nutrients are sensed, the peptides are released and either signal locally to nerves, to other epithelial cells, to muscle cells, to immune cells, but also by endocrine mechanisms, so circulating to trigger effects distally. So these are another picture of endocrine cells. As you can see, just these are CCK secreting cells, but only quite scarce within the gut. They're scattered. They taste the luminal contents, and these are we think there's about 20 different endocrine cell types now described in the gut. So this is just a few of them. So gastrin I mentioned before, acid secretion, secretin released in response to acid in the duodenum to stimulate pancreatic bicarbonate secretion. Cholecystokinin stimulates bile release from the gallbladder and also pancreatic enzyme release. And then, of course, the flavour of the month at the moment is the GLP-1 hormone release from L cells, which is what the various injections to suppress appetite are mimicking. And again, just a few about the complexity of endocrine cells. And I think there's all sorts of things we don't know about these cells. And the problem is because they're so diffuse, they're difficult to study compared with other endocrine organs, which are kind of solid entities, but this is a very diffuse system, and our understanding is really quite limited to what you can measure in the blood at the moment in humans. But these are important because, for example, they activate vagus nerve afferents that signal to the brain, which may control appetite to some extent, perhaps, but also then efferent pathways that go back to the gut. For example, slowing down the rate of stomach emptying to maximize digestion and absorption. And of course, I think for this network, it's going to be increasingly important to understand how the immune system and inflammation interacts with all these physiological processes. So the idea is that the gut is a surveillance system inhibiting food intake with the release of things like CCK and leptin and GLP-1, then also other things such as ghrelin that stimulate food intake. But I think if you dig deep into the literature, convincing evidence that these have got a big physiological importance on food intake in free living healthy humans is actually quite limited. And because I suspect if these were so tightly controlled and effective, then people's food intake will be closely regulated in the way that other physiological processes are. But clearly, humans can choose to disobey these endogenous signals and overeat, and that's probably because the liking and wanting of nice food overcomes the natural endogenous satiety signals that we do have from the gut. And interesting, actually, most of the data that support anorexic effects actually derive from infusing peptides of the hormones into people rather than actually food intake. And to reduce food intake by peptide infusions, you've got to increase plasma levels of CCK by almost fourfold, GLP-1 fourfold, and PYY 3.1-fold by these infusions to actually suppress energy intake. But in terms of eating food, you can only actually achieve that level of increased gut hormones in very few studies. So the idea that release of endogenous hormones in response to food actually suppresses food intake is actually quite questionable, I think. So for example, while CCK does play a role in the acute regulation of appetite and energy intake, there's little evidence to suggest that receptor antagonists or modulations of endogenous CCK by dietary manipulation have any sustainable inhibitory effects on energy intake. Then going back to my original point, why would it really? Because why would you limit food intake at sort of too early? In terms of survival and evolution, we really need to get as much nutrition on board as we can when we have the opportunity. So in a sense, it's not a surprise that these don't have a major appetite suppressing effect in normal free living human beings. That the current drugs, the GLP-1 analogues, are really not replicating physiology at all. They're pharmacological. They've got endogenous GLP-1's only got a half-life of a few minutes. These have got half-life of up to days, and very, very super physiological, effective, but they're not mimicking physiology. So for my last little part of the talk, I just wanted to wonder then if endocrine cell physiology could be perturbed in gastrointestinal disease. And this is a little old, so apologies for that, but I just wanted to put this into the mix for the discussions of the GIBA network going forward. So of course it could be that we in disease circumstances, we know that people lose their appetite, they've got gut inflammation, and could gut endocrine cells be involved in that, was the question. And this was a paper we published a few years ago now, suggesting that yes, it can. So this is looking at CCK in response to a gut infection that affects the proximal gut called Giardia. We've discovered this because one of my healthy volunteers for a study came back with very, very high CCK levels, which was concerning. But then it turned out he wasn't a healthy volunteer at all. He'd got Giardia and was feeling quite sick. So he recruited a panel of more patients and found this was quite reproducible. And in a few patients who let us do it again after the Giardia had been treated, their CCK went back to normal. So the limit to what you can do in humans, but that would suggest that this amplified CCK, sort of pathologically high levels of CCK, that the levels that you get with infusions of intravenous CCK as opposed to release in response to normal nutrients is probably having an amplified effect on the vagus nerve and suppressing appetite that way. So we wanted to take this a little bit further and look at intestinal models to cause small bowel inflammation. And the way we did this was with a parasite worm called Trichinella spiralis. So this is lives in raw. Its eggs live in raw pork, but when it gets into the gut, it causes a transient proximal gut inflammation that lasts for about a week. Then it migrates to other parts of the body, like the muscle, and resides there until the body is eaten, which of course doesn't happen for humans, but does happen for pigs, for example. So trichinella causes this gut inflammation that lasts a week or so, peaks at about a week rather. And when we looked at the food intake of mice infected with Trichinella spiralis, we got this significant fall off of food intake by about 50% at the time that the gut was most inflamed. And then as the gut recovers, the food intake went back to normal, which isn't a surprise. You've got an inflamed gut, but it gives us an opportunity to look at mechanisms. And what we found, just like the humans with Giardia, plasma CCK was significantly increased during the course of the Trichinella induced gut inflammation, and then went back to normal as the gut inflammation receded. The number of CCK cells increased as well, and CCK mRNA went up fourfold during the response to this inflammation. So of course this might be appropriate if you got an inflamed leaky gut, then suppressing food intake until it's better could be biologically appropriate as a response to that pathology. And if there's a CCK blocking receptor drug called loxyglumide, and if we gave loxyglumide to the mice losing their appetite with infection, if we block the CCK receptors endogenously, then the mice ate more, despite the fact the gut was just as inflamed as it was otherwise. And then we also managed to get hold of some CCK knockout mice. So these mice have no CCK cells in the gut. And even though the gut was just as inflamed as in wild-type mice, that these CCK knockout mice, when they were infected, didn't suppress appetite. If anything, they ate a little bit more, despite the fact that the gut was inflamed. So really quite compelling evidence that CCK is driving this anorectic response to gut inflammation. So that really does start to suggest we should be thinking more about gut endocrine cells in the context of inflammation. And interestingly, the CCK knockout mice should be obese if endogenous CCK was regulating food intake, but they were completely normal. So that's just a footnote. And then this is my only bit of proper immunology, that if we gave CD4 T cells from infected wild type mice to immunodeficient mice, that then they actually recapitulated the loss of food intake that you didn't see in the naive immunodeficient mice. So again, it's all in keeping with this being a CD4 T cell driven gut endocrine effect. So back to humans. So probably the best known and commonest disease of small intestinal inflammation is Crohn's disease. So Crohn's disease is an inflammatory bowel disease that can affect anywhere from the mouth to the anus, but very frequently inflames the small intestine. And this is looking at various measures of fullness and hunger in people with Crohn's disease, and you can see that they've got, even before they eat, they've got suppressed appetite, feeling more full and more bloated before eating. And this is looking at gut hormones in people with Crohn's disease. So healthy volunteers, GLP-1, healthy volunteers, peptide YY, and quite significant increases in these anorectic hormones in people with active Crohn's disease, suggesting that some of this appetite suppression that we see in people with Crohn's disease as well as Giardia could well be driven by the gut hormones. This that we chose, these GLP-1 and PYY mainly come from the distal small intestine and the colon, which is where Crohn's disease more commonly is found. Crohn's is quite rare in the duodenum, so we didn't really have patients to look at CCK in enough detail. So wrapping up maybe a little earlier, I'm giving you hopefully an overview of gut function and structure, which fairly basic level, but hopefully for those of you who are new to the gut field, that'll give you some orientation for what we're going to focus on in the future. Of course, my colleagues will add in elements concerning the microbiology, microbiome, the neuroscience and the immunology. But just again, a bit of a plea to consider the under-researched endocrine system in all of this and the vagus nerves, and finally to make sure we consider this across the whole life course, not just in particular parts of that. So there's my email address if anybody wants to get in touch after the talk. The picture in the middle is actually a knitted human gut that was given to me when I was the chair of the charity called Guts UK, and when I sat down, they kindly awarded me this complete knitted gastrointestinal tract from top to bottom, which always goes down well at meetings. And with that, I'll close my slides, and I'm happy to take any questions.
Thanks, John. Very nice overview. Thanks very much for doing that. So as I mentioned in the chat, feel free to ask any questions you may have. I'll get started with one while people think of their own. John, so I guess I wanted to get your take. So, you gave some examples of looking at these GI function in other animals, such as, mouse with your experimental models. But I want to get a bit your perspective on what do you think is the best approach now if we have a hypothesis about some function in the human GI tract? What are the best models? Because there's a bit of a push to try to go away from animal models, right?
Yeah.
From the UK government in general. So what is your opinion on that? Do we have good in vitro alternatives to mice? Are other mammals potentially a better representative of the GI tract in humans? What's your perspective on the experimental approaches we should use?
Yeah, and I completely agree. Ultimately, if we can, what we can do in humans is best. So I guess the three possibilities that imaging is getting better and better. So our ability to image gut function. So things like MR scans with very rapid acquisition of images that before gut movements and peristalsis made MR scanning of the gut very difficult to do, but that's really advanced now. So I think looking at those as, and we can measure things like transport through the gut using those and various other techniques. I think organoids, taking biopsies from the gut and growing them in culture are much better than cell lines, obviously, and that gives you the opportunity to put different components in. But the problem with the organoids, they generally lack the neural tissue, so you can see what's going on in the epithelium and superficial layers, but you can add neurotransmitter analogues and antagonists and so forth, so you can still do that. So I think the human-derived organoids are probably going to be the biggest strength going forwards. Brain imaging as well as gut imaging, because I think a lot of this, if we know that I didn't show in the talk, but we've done quite a lot of functional brain imaging showing activation of brain areas in response to nutrients put in the gut. So I think that's been main. So using human tissues, and then really old-fashioned things like obviously just surgical samples for more gross things, but then that tends to be pathological, of course, because people don't have healthy gut removed; they only have diseased gut removed. So in terms of physiology, I think that's limited. So I think organoids are going to be the big thing.
Yeah, and so I guess when we analyze results that come from mouse models, so what are the main differences you would expect between the GI tract of a mice versus a human, and what could lead to potential, misleading results and misleading interpretation? Do you have any sort of ideas on that?
Yeah, I mean, they are a different species with different immune system and different transport mechanisms and so forth, but they do seem to replicate most aspects of gut function well enough for that. But again, there's a limit to how far you can go with mouse models. I mean, for example, they're nocturnal; they don't eat in the day, so that's completely reversed. We know that the gut's got quite strong circadian rhythms, even at the epithelial level. So some mice are clearly not things like food intake and appetite regulation. Something that eats completely out of phase with humans can't be the best model.
Yeah, and what do you see as kind of the main gaps in terms of our understanding of the function of the GI tract? So what are the things that you think we still don't know a lot about that actually we should investigate a bit further?
Yeah, there's a lot. I mean, the big one for me is just how much variation there is in gut physiology and anatomy. So the most physiological systems are much more tightly regulated, and things are similar in size and scale and things. But gut function, you can have somebody who has normal gut function that moves the bowel 5 times a day, and somebody who goes 1 time a week. And I sort of think for me, one of the big questions is how we understand variability in the human gut, and to what extent that matters. So from a clinical perspective, we're not really interested in the baseline. It's only when there's a change that we need to do that. And then I think the other big thing is the whole kind of what's happening in vivo in life at the epithelial level.
Yep.
How to sample things in the gut. So you can get things from the esophagus and stomach fairly easily, and from the colon very easily, but it's very hard to get samples from the small intestine. Although some interesting work with Gary Frost's group in Imperial that are putting tubes in the whole length of the small intestine. Cool. And they're placed under radiological control, and they can get them right into the distal ileum and take samples from there. So I think it's those dark corners of the gut that we've had limited access to, where the mystery is. And of course, that's mainly the small intestine, and the small intestine is where most of it happens. So I think that would be my biggest hole would be the small intestine.
Okay, so we have a couple of questions now in the chat. So the first one is, what are the most recent breakthroughs in the GI tract gut immune brain axis, and what are we likely to discover in the near future? So where's the research heading?
Yeah, I'm not sure there's been that much in the gut immune brain axis. I guess that's the reason this network is required. So I think I'll throw that question back to you guys with you. You know, that's what we need to do next. None of this actually made any clinical impact yet, but I think ultimately that's where we'd like this to go. That we can. I think in terms of the microbiome's not my area of work, so I'll probably park that question for one of the future speakers whose expertise in the microbiome.
Yeah, we do have another one on FMT, on fecal microbiota transplants. I don't know if you want to answer that, or we'll leave it to Leslie. But they are asking essentially just your opinion on FMT experiments to learn about host microbe interaction. So in the context of reducing animal experiments, so we're talking about, not being able to do experiments in humans, but fecal transplants potentially could help with that.
Yeah, they could, absolutely. So we know clinically they're quite effective in some situations. So things like Clostridium difficile, which can be life-threatening, FMT can be life-saving for those, whereas it's been quite disappointing for things like inflammatory bowel disease. Crohn's and colitis haven't responded terribly well, although it does seem to be that there's particular donors have that. I think the ethics would be difficult. I think obviously it's doing it as a clinical intervention in somebody with a disease makes sense, but because there's obviously risks of transferring pathogens and other adversity, I'd be surprised if we got to the point of doing FMT in healthy humans as an experimental thing.
Yeah, it's risky.
So I think that would have to go back to the animal model and all its caveats. But maybe those people who are having FMT for clinical indications, maybe we need to do more in terms of. I think it's a great work going on in Birmingham around that at the moment, and of course probably we'll get to the point where when we know what's working in FMT, it'll become synthetic. So rather than having to collect actual human feces and put them down a tube, growing that in a lab and giving what's necessary to repopulate the microbiome must be the future direction of travel. I think actual fecal transplant will be. When we look back on this in 20 years, it'll seem a bit of a crazy thing to have done, but
it's still a bit of a blind exercise, I feel. And people, they screen for pathogens and potential harmful bacteria, but then we actually know very little what we're doing with the FMTs, what we're actually putting into different people.
Absolutely.
Okay, we have another question. So this is also a bit microbiome related, but they're asking more about, so what are the big unknowns in terms of microbiome-mediated regulation of gut barrier integrity? So what are the best reliable markers used in the clinic for gut barrier integrity?
Yeah, so we don't actually do gut barrier integrity in clinical practice at all. It's entirely research. But you can do things where you put things like inulin in the gut and measure it in the urine, or chrome EDTA and things like that. But they don't actually have any clinical usefulness. So there's plenty of methods out there for research purposes, but it doesn't, at the moment, it doesn't have any clinical usefulness. So we don't actually do any of them in actual clinical practice.
Mhm. Another question. So what are the key disadvantages of using an immortalized cell line in 3D culture to make an in vitro model over using human tissues to make an in vitro organoid model?
Yeah, it's not something I've worked on myself, but I guess it's the fact they are immortalized cell lines generally from tumors means that there's something wrong with them. Really, they're not normal cells. So I think, whereas if you can use primary actual tissues from healthy or diseased states, then that must be better than using a immortalized cell from a mouse tumor, for example. They've got their place, but using primary tissue from the right species must be better.
Yeah. Are there new in vivo technologies that are able to track the GI in real time, such as the pH?
Yeah, that's really good. So we can do pH anywhere in the esophagus and stomach, so that's quite easy to put probes down and measure for 24 hours, which we do as a clinical thing. There was something called the smart pill, which was a little capsule device that was swallowed and it go through the small intestine. It measured the pH through the whole small intestine and did manometry to measure contractions and function through the whole small intestine. And that was marketed but had such poor clinical take-up that the manufacturers discontinued it. I don't know if they still have a stock that can be accessed for research purposes, but they took it off the market because it was not really useful in clinical practice. It didn't really catch up. So the technology is there, but just not on the market.
Okay, thank you, Professor. I have a question. Which do you think holds more promise for future metabolic therapies, targeting the endocrine cell directly or modulating the neural pathways that transmitted signals from the gut to the brain?
Yeah, I think I sort of alluded to this. I think it's stimulating the gut hormones directly to release the endogenous enzyme hormones actually has quite a minimal effect on appetite regulation, whereas the things like the GLP-1 agonists that we have now will be working on the endocrine cells. They're working on the gut to brain signaling pathways. So I think modulating the neural pathways and mimicking those is always going to be more potent than. So I don't believe that endogenous gut hormones do very much in terms of appetite regulation. It's the actual human data for that is quite poor, as one of my slides hopefully described.
Excellent. So if the GLP are not mimicking the normal GI function, what are then severe possible long-term repercussions for those who are using these?
Yeah, we'll find out soon, I imagine.
Yeah, exactly.
Yeah. There's already a bit of a signal of causing increased risk of pancreatitis. So that seems to be already happening. That we're seeing pancreatitis in people with these agents. My other worry is that people get them who really don't need them. So people are using them as people have got misperceptions of body image and actually underweight and malnourished, managing to access these drugs and making themselves more underweight and more malnourished. So that's one of the bigger concerns. We don't actually, it's endocrinologists rather than gastroenterologists who actually do the management of these cases. But we are seeing people who come along with some vomiting and nausea and other predictable side effects because they slow down gastric emptying quite a lot. And often they refer, and people haven't joined the dots and realised that the nausea and vomiting is actually caused by the GLP-1 supraphysiological agonists, which some people seem to be more sensitive to than others. I guess it'll be a trade-off on it, whether how much the reduction in diabetes and cardiovascular disease and other complications, how it will hopefully be quite a major gain, but it's a question of how big the downside of things like pancreatitis will be and whether we'll be able to predict whether certain individuals are more at risk than others, for example. But it's a big experiment going on in the real world at the moment, and time will tell.
Yeah. And then last, there's more of a general, I guess, question. Interrelationship between gut peptides, microbiome, their impact on appetite under severe enteritis. Not sure if you
Yeah,
have any thoughts on that.
yeah. I think my CCK example shows that it's almost certainly going to be a thing like so enteritis. So the worms or Crohn's disease or Giardia all cause enteritis, and I think the evidence is quite strong that it's the release of gut peptides is driving that reduction in
Mhm.
appetite, which is probably an appropriate response biologically. I suspect it's probably not as soft human beings. We don't like feeling poorly or sick or whatever, but probably in reality, if you've been drinking river water that's full of parasites, you probably stopping it for a few days is probably a good thing to do. So I think these are probably adaptive responses rather than just obviously different in things like Crohn's disease where things weren't. That's a pathological situation that you don't need an adaptive response to. But I think in kind of environmental and evolutionary terms, these are probably appropriate responses.
Great. Then we have another one. So from your perspective of gut biology, what are the most important cells to include in an in vitro model and the proportions of each cell type? For instance, enteroendocrine cells are scattered and are less frequent as compared to other cell types in the gut.
Yeah, yeah. Well, I guess if you do organoids, they should be there in the right proportion already. Would be the thought. But if you're doing it in a sort of adding things in, I guess we know that 90% of the cells are enterocytes and about 5% are goblet cells and 1% are endocrine cells. So I guess you'd want to recapitulate that as closely as possible. But if you use biopsy derived organoids, then you should have the right mix already from the outset.
Okay. And the last one is, as we learn more about the aging gut and its role in inflammation, how has that changed the way you approach functional gut disorders in older patients compared to younger ones?
Yeah, that's a really interesting question. There's at the moment it probably doesn't change the approach. Although I think people used to think that older people didn't really get functional gut disorders, but that's just not true. So, but things like constipation and slow transit certainly get more common as people get older. But I'm not sure there's much of an inflammation aspect to that. That's probably just kind of poorer function as time goes along. So at the moment, I don't think it's made any specific differences, and we tend to treat everybody the same. But maybe that's not the smartest way to do things, and we'll learn more as time goes on.
Excellent. And then we have, what do you think the role of bile acid metabolism in the regulation of gut hormones?
Yeah. There's not been much on that I'm aware of. There's a little bit of evidence that bile might suppress CCK secretion, but I'm obviously not much bile gets into the colon physiologically. It mainly gets reabsorbed in the small intestine, but some bile acid does get into the colon, and we know that is metabolized to different bile acid derivatives in the colon. There's a condition called bile acid diarrhea when you get quite a big overflow of bile into the colon because it doesn't get reabsorbed in the terminal ileum as it should be. And there's a couple of papers showing how the microbiome is quite different in people with bile acid diarrhea. So rather suggesting that bile acids do have an effect on the microbiome if the microbiome is exposed to them, but the colonic microbiome doesn't really see much bile acid in healthy people.
Okay. I have one more question out of curiosity. This might be a bit naive. What is your opinion on the appendix? Because there's obviously a lot of talk about, historically people thought that the appendix doesn't do anything, and obviously a lot of people don't have them because they need to take it out. But then there's a new wave suggesting that it can potentially reseed the microbiome. I think this seems a bit more, far-fetched, but I wanted to get your perspective as a more of a clinician. So what do you see as kind of the role of the appendix in the gut?
Yeah, I don't know. Yeah, it's mainly the surgeons who deal with the appendix rather than gastroenterologists, so we just tend to ignore it really. And you're right, it can't, given that you can chop it out with pretty much total impunity. I don't know if anybody have done. It'd be interesting because obviously with things like in the UK Biobank database, there must be an awful lot of people who've had appendicectomies. And I just wouldn't. And if anybody's actually looked at their downstream likelihood of developing other disorders, would be a. I'm going to look that up after this talk, because a giant database. You don't. There's a little bit of evidence that people having appendicectomy are less likely to get inflammatory bowel disease, but so it's possible, but that's quite weak. But it's the same as the gallbladder. Really, you can chop the gallbladder out with, and most people have no problem. They don't become malnourished, and again, it's probably because we're living in an excess of food. So if food was hard to come by, then probably not having a gallbladder and bile with food would reduce your digestion and absorption. But because we've got largely overwhelmed with an excess of fat, it doesn't matter if you can't digest it quite as well, because you'll still have plenty of it.
Yeah. We have one last one. Thank you, Professor, for the great talk. Given the ENS and the GALT are in constant crosstalk, how much of the brain signal is actually just a downstream effect of macrophage in the lamina propria?
Gosh, yeah, this is really, that's too immunological for me as a poor clinician, I think.
It does seem a bit technical.
Yeah, but maybe it's a good thought about maybe just one. Maybe not just macrophages. I guess there's so many other immune cells in there, but I really wouldn't know that. But again, it's down to what the role of this network is, isn't it? Understanding these crosstalk between these things and getting the right models is. So that's probably a question to ask again in 3 years when we know more.
Yeah, we might get a bit more clarity on that on Thursday at the immune session. Okay, I think that's probably a good time to wrap things up. So let's thank again John McClellan for the great talk.
Thank you.
Thanks, John. And now we're going to move on to the second speaker, Leslie. I assume you're there. I can see. Yeah.
Leslie. Yeah, I'm here. Let me just share my screen.
Excellent. Thanks, Leslie. Okay, so Leslie is a professor of microbiome and systems biology at Nottingham Trent University, and she focuses on microbiome research. Her research combines both microbiology and bioinformatics approaches, which is something that is also actually quite rare in the field, in vitro and in vivo models to understand how members of the gut microbiota function that influence health and disease.
You need to give me permission. Sorry, you need to give me permission to share my screen.
Sorry, yeah, I'll do that.
Sorry, Alex, to interrupt.
Yeah, no worries. Let me see. Oh, you seem to be. I think you have it. You're a presenter. You're labeled as a presenter. Does it say you don't have it? Maybe try just reconnecting. You're at least in the list here. You're labeled as a presenter, so you should have access.
I'll just do it on my entire screen and see if that works.
Okay.
Can you see that?
Not yet.
Here we go again.
Teams.
Yeah. Why is it not letting me share?
Do you want to try just restarting? Maybe rejoining?
Yeah, give me a minute. Sorry about this, folks.
No worries. That's okay. We have a couple of minutes. That's okay. Yeah, so as I was saying, so Leslie has specific expertise in anaerobic microbiology and the processing of various types of omics data, so microbiomic, transcriptomic, metabolomics, and genomic. And so her talk is going to be focused on the gut microbiome specifically and giving us an overview of the field and what things to look out are. Oh, perfect. Yes. Now. Excellent. Leslie, I don't think we can hear you. You're muted, but we can see the screen now.
Yeah, but that's not the screen I want to be shown. I'm not quite sure why. I might have to send you my slides, Alex.
Which one do you see? I'm seeing the screen like full screen. I'm not seeing any issues.
But that's host microbiota interactions in pancreatic cancer, isn't it?
Yes. Yeah. Okay. You don't want to share those slides. Okay. You want to know.
No.
Yeah, whatever makes it easier. I'm happy to share from my side if you're not able to do it on your end.
Yeah, I can't do it. So give me a minute and I'll send you the slides.
Okay.
I'm sorry about this.
No, that's fine. That's okay.
It's because my Mac is due for scrapping.
Okay.
And it won't let me share things on Teams anymore. I think that's what the issue is.
Okay.
Should have done this from home.
Should be okay.
Right, I've just sent it to you.
Okay.
Thank you everyone for your patience.
Let me. You send it through mail? By
Yeah.
mail? Okay. Then yeah, refreshing, see if it gets here. Just bear with us, everyone. It should be quick. Might be a big slide deck. That's why it might be taking a bit longer. Do you think it's potentially due to the size of the PowerPoint slides?
I have
that you're
no
unable to share that
idea.
one?
The other option I've got is I can stick it on Dropbox and see if I can share it via there.
Still haven't received. I assume it's because it's bigger ones, so it might be taking a bit longer to upload.
I'll just see if I can get it on Dropbox, and that might be the easiest way of doing it. I apologize everybody for this.
Yeah, that's okay. We have a bit of time, so I think it's okay.
Right.
Okay.
You can
Yeah,
see that.
we're seeing it. Yeah, now we can see it. Yeah.
Right, excellent. So yeah, thank you everybody for your patience, and I apologize. Yeah, I apologize for the delay. So as Alex said, I'm a microbiologist originally. I did my PhD at the University of Reading with Glenn Gibson, and then after going to APC in Cork to work on bacteriophage for a bit, I retrained as a computational biologist and bioinformatician. So I've worked across several different fields relevant to the gut-brain axis. So I just want to start, because I'm coming at this as a systems biologist. I want to explain the difference between the microbiota and the microbiome, because the two terms are used interchangeably in the literature, and they do actually relate to different things. So when we're talking about a microbiota, we're talking about all of the microbes in a specific environment. So that includes the viruses, the bacteria and archaea, and the eukaryotes. When we're talking about the human gut and the human body in general, you find lots and lots of viruses, you find lots of bacteria and archaea, and you find a few eukaryotes. So in terms of the human gut, you're looking at greater than 10 to the 12 viruses, maybe the same number of bacteria and archaea, but because it's so difficult to quantify viruses, we don't know, but they're there in very high abundance. And with the eukaryotes, they're just not there in very high abundance in the human gut. You occasionally pull them out when you do isolation work, but to characterize them in detail, you need really specialist methods adapted specifically for looking at things like fungi, protists, worms, any other eukaryote you can think of. To put it in context, that picture on the right-hand side, the big pink blobs are human blood cells. The big purple circles are yeast, and then you've got the thin rods are bacteria. So that's to try and give you an idea of the size of these things. You can't see viruses under a normal light microscope. You can only see them under an electron microscope, which is why, again, they're very difficult to study in detail. But as far as the viruses are concerned, pretty much everything in the human gut is a bacteriophage. So that is either a virus that kills bacteria or a virus that integrates into a bacterial genome and has the potential to be released into the environment at some time in the future, usually a stressor, something like antibiotics. But by far and away, most bacteriophages present in the human gut are ones that integrate into bacterial genomes, and they're not necessarily ones that kill other bacteria. So when you think about the microbiota, you're talking about the entire human body has bacteria within it and across it. So every surface has a microbiota. Your nose, mouth, lungs, colon, vagina, skin, everything has a microbiota. And they're there generally to try and help you, protect you, or to interact with your immune system. So it's very much a two-way communication. But you will see from this picture on the left that the different regions of the body, every region has a different composition of microbiota. So as far as the gut microbiota is concerned, we're talking about everything from the esophagus, stomach, small intestine, down to the large intestine. As John explained earlier, it's pretty much everything from the esophagus to the anus that we're interested in from the microbiota. And you can see there are big differences between the esophagus, stomach, small intestine, and large intestine in terms of the dominance of bacteria. So in the large intestine, it's mostly Bacteroides, Firmicutes, so Clostridia, really obligate anaerobic bacteria that you find in feces, whereas the small intestine, you have far more proteobacteria, far more facultative organisms. So basically organisms that will grow in the presence or absence of oxygen. And this massively undersampled. Similarly, the stomach, we know lots about Helicobacter, but we don't know much about what else is in the microbiota in the stomach. And the esophagus is only really just being started to be looked at, and I'll present some data on that later. As I said, we're talking from the gut microbiota, we're talking from the esophagus to the anus, and you can see looking at this figure that the environmental conditions throughout the intestinal tract change. So it's not just pH changes, it's also oxygen availability that's changing as you go along the system. So the esophagus tends to be aerobic, basic environment. You get down to the stomach, as John alluded to, it's an acid bath. It's designed to destroy pathogens, but that doesn't mean that there's no bacteria in there. They still can survive. You get to the small intestine, you've got a few thousand bacteria starting to be present in samples, to 100,000 to 1 million sometimes by the time you get down to the ileum. And then you get to the large intestine, the proximal colon, so the ascending colon, the bit on the left-hand side of this image, that's the most densely populated ecosystem on the planet, and that's where most of the fermentation of fibers and things occurs. And then we use feces as a proxy for what's going on in the gut. That fecal microbiota is representative of feces. It doesn't represent what's going on the different regions of the gut. And that's something that we need to get better at understanding, is how these different communities across the entire system interact with the host. So this is quite a nice infographic put together by Gut Microbiota for Health, showing the factors we know them currently about the microbiota. So for a very long time, everyone said there were 3 to 1 human cells, bacterial cells, to human cells, but it's actually closer to 1 to 1 now from the work from Sender. John's already did a lovely discussion of the size of the intestinal tract, and it's important to recognize that fact that if you open up the intestinal tract, even though it looks like a long tube, once you open it up, it's actually spread across a very large surface area. So there's a huge scope for interactions of the microbiota across your entire intestinal tract. Everybody's microbiota is different, which makes working with humans something of a nightmare when you're doing studies where you're looking at interventions or clinical aspects, because there's so much variability. And it's also important to recognize as well that not everybody responds in the same way to treatment. So we've got this concept of responders and non-responders. So working out why some people act with why some people's microbiota or medications work when others don't, because of this responder non-responder. And that's something that's going to become more and more important as studies advance beyond just doing 16S and sequencing studies. The important thing to recognize as well is, from a genetic perspective, you are more bug than you are human. There are 150 times more bacterial genes in your gastrointestinal tract than there are encoded within the human body. And that means there's massive scope for the microbiota processing processing things that you eat or ingest in ways that your body cannot do. And that is what has become apparent, is that the biological capacity of the microbiota is more important possibly than whether or not specific groups of bacteria go up and down in abundance. And remember as well that you've got 10 million bacteria across your entire intestinal tract, and the kind of work that Alex has done in terms of metagenome assembled genomes and characterizing the diversity of the microbiota has been all focused at fecal samples. We do not know what the full capacity encoded across the entire intestinal tract is in terms of the bioprocesses carried out in different regions of the intestinal tract. Now, this is an old figure, but it's still a nice one because it highlights nicely how the microbiota changes across life. So you're probably fully aware that breastfed infants have very different microbiotas from formula-fed infants, and breastfed infants have lots of these, what we call actinobacteria at a phylum level, but actually at a species level, it's mostly bifidobacteria. And because those bacteria are essentially designed for digesting human milk oligosaccharides and components of human milk, formula-fed infants obviously don't get that exposure from maternal milk. So their microbiota tends to be more diverse. So there's lots of push in industry to try and make prebiotic formulas that are more like breast milk so that you can give children the best start in life as possible, because we know that you can see differences in terms of allergic responses in formula fed, the kids who were formula fed up to 7 years of age. So they're much more likely to have things like atopic dermatitis, eczema, if they were formula fed as children. So what happens in early life in terms of microbiota has consequences later in life. In terms of what happens in toddlers and adolescents, we really don't have much data there at the moment. That's something that's working on, and there's big studies that have just finished on human exposome projects in Europe looking at adolescent and childhood cohorts, and there's going to be lots of information coming out on those soon. So that's something to bear in mind for the future. This figure originally said that it was healthy versus obese individuals. I've deliberately changed it to metabolically healthy or metabolically unhealthy because it's clear that you can be obese and there be nothing physically or metabolically wrong with you. It's just the way you're made up. A bit like John saying, there's lots of differences in intestinal transit. There's lots of differences in terms of the manifestations of obesity and the effect it can have on the host. But what's become apparent as we've extended studies to type 2 diabetes and cardiometabolic diseases is that there's a very big difference between metabolically healthy and unhealthy individuals in terms of their microbiota and the effects that has on the system, or the effects that systemic changes on the host is having on the composition of the microbiota. It's still very much a chicken and egg story at the moment in terms of which one's driving the changes. But the important thing to acknowledge in metabolically unhealthy individuals is they tend to have lots more of these proteobacteria, so things like E. coli, Klebsiella species. And all of these bacteria have LPS in their cell wall. This is just for the benefit of the non-microbiologist in the room. So you've got a gram-negative bacterium cell, then they've all got this lipopolysaccharide, which is this antigen and outer core. In animal models of obesity, this LPS has been shown to cause metabolic endotoxemia, so to contribute to low-grade systemic inflammation, but that has not necessarily been translated to human studies, and that's something that needs looking at because it's clear that these changes in the microbiota cause leaky gut, or leaky gut preempts the changes in the microbiota, but the circulating effects of that in terms of LPS means that potentially in humans you're also getting this metabolic endotoxemia, and it needs to be far better studied because it really hasn't been looked at in as much detail in human cohorts as it has been in animal cohorts. So I'm coming to this as a systems biologist. So what tends to happen when people are working on certain aspects of the microbiota is they're interested in only one bit. They're interested in the brain, or they're interested in the gut, or they're interested in the lungs, the liver. There's lots and lots of associations of the microbiota and its composition with different diseases. And it's a bit like the Daily Mail blaming everything on cancer. We're currently blaming everything on the microbiota. But if you look across all of these different conditions and diseases, you see the same trends in terms of the changes in the microbiota. You're seeing in loss of the specialist organisms, so the really obligately anaerobic bacteria that can produce the short chain fatty acids that we know are associated with good health. You're seeing an increase in these facultative organisms, so the ones that don't like, the ones that will grow aerobically or anaerobically. So you're getting lots of bacteria that are very similar to one another in terms of their genetic function compared with a rich, diverse microbiota that you see in metabolically healthy individuals, where they have a much higher what we call microbial gene richness, so counting the number of unique genes that the microbiota encodes, you see much higher levels in metabolically healthy individuals than you do in anybody that's got any kind of inflammation-associated disease. And you may think it's a bit odd to include Alzheimer's here as an inflammatory disease, but it's actually been called type 3 diabetes in some instances. So there's clearly a link with inflammation and what's going on systemically in the microbiota. Now, I've deliberately called, when I'm talking about all the different bacteria, the microbiota. If we look at the microbiome, as a systems biologist, I'm talking about you and your microbiota. So it's very much a two-way interaction. And I think sometimes because of the focus on the microbiome in the literature, we forget that it's not just a bacteria, it's you and the host. Everything that you ingest, everything your body produces will have an effect on the microbiota and its composition, which will in turn have an effect on your host because it's constantly signaling. So you've got the living bits, which are all the organisms, except for the viruses, because philosophical whether or not you consider them living or not. But basically, the bacteria, archaea, and eukaryotes will interact with the host, and similarly, the host will interact with those organisms. And then you've got all the abiotic factors as well, so the non-living bits that can influence the living bits. So that can be the surrounding environmental conditions, it can be pH, oxygen availability, water availability, which John mentioned was really important in the colon, temperatures, all those secretions, antimicrobial peptides your body produces, the nutrient availability in the different regions of the gut. Everything has an effect on the microbiota. And you can see there are many intrinsic factors that will affect the microbiota. Sorry, I'm just having a drink. There are many intrinsic and extrinsic factors that affect the microbiota and the microbiome. So the abiotic factors, the non-living bits, as you've heard previously, gastric acid, motility, lots and lots of different immune factors that your body secretes. And then you've got everything, the extrinsic things, and things you put into your body, diet, prebiotics, antibiotics, laxatives, non-steroidal anti-inflammatories. Plus, we know it clearly as well, physical activity, metabolic health, age, sex, hormone status, everything can have an effect on the way that the microbiome influences host health. And again, to reiterate, 99% of the genes in your body are of microbial origin and not of human, and that massively increases the way in which your body is subject to the effects of the microbiota. So, you know, hopefully all of you know that dietary fibers, you ingest dietary fibers, the microbiota in the large intestine converts those to short chain fatty acids, so acetate, propionate, and butyrate. Huge amount of work was done on that by the functional food industry because they wanted to make things like bran flakes and porridge more butyrogenic, because butyrate is obviously associated with good colonic health. A huge amount of work was done in that area for a very long time. We've known as well now the dietary methylamines are important, polyphenols. All of those dietary compounds, the way that the microbiota interacts with them will affect whether or not they're bioavailable, whether they're present in an efficacious dose, whether they're potentially toxic, whether they're likely to increase your disease risk. And it's becoming apparent that we need to look at these metabolites that the microbiota produces from what we ingest. We need to look at them in a more pharmacological context. So what is an efficacious dose of these things in the circulation or in the gut? What is a null dose? What is a toxic dose? And it's only that way we're going to understand how the microbiota is affecting things, not just at an intestinal level, but also systemically. And you may think it's a bit odd to talk about the microbiota and systemic health, but what happens is you ingest all these different things. The microbiota in the small and large intestine can act on these medications, foods that you ingest. Those metabolites can be taken up unchanged to the liver, and it's important to remember that 75% of the blood supply to your gut comes directly via the hepatic portal vein. So there's lots and lots of microbial metabolites and components that your liver is exposed to, even if you never see a bacterium in your liver on a daily basis. And those metabolites can enter the circulation, and then they can interact with the brain and other cells throughout the body. So short chain fatty acids enter the system unchanged. You don't tend to see lots and lots of butyrate in the bloodstream. acetate and propionate can get into the bloodstream. So in the human gut, these metabolites are present in 100 millimolar levels. Acetate 100 millimolar, about the third of the amount for butyrate and propionate. But when you get into the circulation, they're only there in about 1 micromolar concentration. But that doesn't matter because they are almost perfect ligands for the free fatty acid receptors 2 and 3, which they can act upon and affect things like gluconeogenesis, mTOR signaling. We know that FFAR2 is present along the blood-brain barrier, and that propionate, for instance, can interact directly with the blood-brain barrier. So these things are having a systemic effect. So it's not what happens in the gut isn't staying in the gut, and it's important to acknowledge that in studies when we're looking at the gut-brain axis. And the other important thing to say as well is in relation to the short-chain fatty acids, we know lots and lots about the bacteria that can produce short-chain fatty acids from different substrates. And this just highlights what was more or less recent knowledge. It needs updating with things like myo-inositol from the work that Patrice Canny's just published. But basically, this is still the only group of dietary substrates that we have a real idea of the bacteria that actually do break down substrates to specific metabolites. And you can see it's incredibly complex. And most of this work was done at the Rowett Institute by Sylvia Duncan and Petra Louie. And I suppose I included this to highlight that we need to do some old-fashioned microbiology and biochemistry, because when you look at the metagenomic data and functional data we generate, currently we can only annotate 30%, up to 30% of genes in studies where we're looking at metagenomic data, looking at the functional diversity of the microbiota. And unless we properly discover what most of the genes in the human gut microbiota are doing, we're never going to get a proper handle on how we can manipulate these things better for human health. So I said that the metabolites can enter the human enter circulation unchanged, or they can undergo phase 1 metabolism. So phase 1 metabolism can occur in the liver and in the large intestine. And basically your body's just trying to modify metabolites to make them more soluble, water soluble, so they can be excreted in urine. It's fairly straightforward. So the one that you may have heard of is trimethylamine N oxide. That's basically an excretion product. Trimethylamine from the gut microbiota is converted to trimethylamine N oxide in the liver, and then your body tries excreting it. You've also got phase 2 metabolism, which is a bit more complicated. And this is basically the way that things like paracetamol and ethanol and morphine are processed by the body. So you don't get rid of the original compounds, you get rid of their conjugated compounds. So glucuronidated or glycated or sulfated products. And the major metabolites that we know something about in terms of the microbiota are hippurate. So that's one of the main components of human urine, is hippurate, and that's produced from the benzoate that bacteria produce from dietary substrates. You just add a glycine onto it, becomes hippurate, and then you can get it out of the system. And then cresol, produced from phenylalanine and tyrosine metabolism by the gut microbiota, that's sulfated or glucuronidated to make them more soluble. And the important thing with some of these metabolites as well is you can't detect all of them easily in human blood. A lot of them are bound to plasma proteins, and that's how they keep protected from the human system. It's only when you have things like chronic kidney disease where you can't clear these metabolites from the system that they start building up, and that's when they can cause problems. So again, it's the context of this metabolite may actually be beneficial or not having an effect in a healthy individual, but it might be having a big effect with somebody when they've got a disease or reduced renal function. And again, it's thinking about the context that you're looking at these things in. Don't just think, oh, one metabolite bad because lots of people say it is. It's far more nuanced than that. In the same way that there are no good or bad bacteria, it's the same way applies to metabolites produced by bacteria. I included this figure. It's an old one, but it's a nice study. It was one of the first to show that the microbiota and its metabolites are important. So basically, Zheng et al. Fed rats antibiotics. So before they fed the antibiotics, they looked at the metabolites that were in urine and feces. And after they'd fed the animals antibiotics, they looked the same again. And basically, this is all the pathways of a rat. So the zigzag bit on the left-hand side, that's basically fat lipid metabolism, which we still don't know much about, mammalian or microbial. But everywhere on this graph where there's a red dot, that's somewhere in a mammalian metabolic path, all metabolic pathways where microbial metabolites can interact. Now this is 15 years old, this figure. The red dots are probably far increased now, but it's to highlight that the microbial metabolites can have a great impact on lots and lots of different biological processes, and they will differ between every organ or cell type you look at throughout the body. So don't try and infer that results from one organ match another organ, essentially. And again, to highlight, there's hundreds of metabolites that can be produced by the microbiota. And this paper from Han et al. From 2021, where they basically tried to catalog as many microbial metabolites as possible. We're in the hundreds now, and working out how each of these interact with the system or mixtures interact is incredibly complicated. The other thing as well is that we know now from studies done by Tom Wilmansky that you can take 40 metabolites from human plasma, and 13 of those 40 are microbial metabolites, but they actually correlate really well with alpha diversity. We know that studies looking at fecal samples are really difficult and to reproduce across labs, whereas blood studies and blood metabolites and their profiling is much simpler. So it may be that in future we move towards using the blood metabolome as a readout for the microbial diversity and only go and study specific samples we're interested in, just to try and make things more producible and more high throughput, because nobody wants to work with feces if they can help it. It's smelly, it's messy, it's not nice. Whereas it's much easier to get blood and get people to consent to give blood samples as well across lots of different cultural settings. So it's apparent from this work as well from Wilmansky that there is definitely a correlation with this metabolites we're seeing in blood and the microbiota and its diversity. And this is something that's going to be more important with work that we've got coming out that I'm not going to talk about today in terms of metabolic richness as well as microbial gene richness. So yeah, I've been talking about the gut microbiota and the metabolites. We know that there's the gut-liver axis. There's a lot of work being done on looking at the microbiota and fatty liver disease, insulin resistance, cardiometabolic disorders. It's a lot of the work that I'm involved with, and also some of the metacarditis studies that have been looked at across. And the thing is as well, to demonstrate here, whereas those figures I showed earlier, it looked quite linear in terms of you eat things, your microbiota modifies it, it gets put into the system. You can see it's far from linear. So you've got things like enterohepatic recirculation. As John nicely said, the bile acids, they're taken up. So they basically pushed by the body into the small intestine. The microbiota act upon the primary bile acids. They're converted to lots of secondary bile acids that can interact with things like FXR receptors, aryl hydrocarbon receptors, but then they're taken back up in the small intestine. But they will have an effect on the liver. You've also got the portal circulation, as I've mentioned. Urea and ammonia are also subject to enterohepatic recirculation. And then you've got all the other things the microbiota interacts with as well. So the inflammatory cytokines, the chylomicrons. It's a hugely complex system, the gut-liver axis. If you look towards the gut-brain axis, again, you've got this bidirectional communication, but again, the same applies. There's lots of studies identifying associations, but the actual mechanistic data are lacking. We know from work that Philippe Devada did that you can affect insulin signaling and gluconeogenesis by cutting the vagus nerve, because propionate and propionate and butyrate can't act. We know there's lots of immune cell stimulation. We know that different neurotransmitters are produced in the gut. Whether or not GABA and things like that get from the gut into the brain is another matter that still hasn't been resolved. And the work that we're mostly involved in is looking at the effects of these metabolites that get taken into the blood and how they affect the blood-brain barrier, but also cognition. And shameless plug for the work that we're doing in this area with Simon MacArthur, a very long-standing collaboration. But the thing I want to highlight is it doesn't matter whether you're looking at the gut, the brain, or the liver. We know that there's a gut-kidney axis. We know there's a gut-heart axis. There's every kind of axis around. So can we just start looking at it as gut and body and looking at things holistically, because as a systems biologist, I'm fairly agnostic as to the system I'm looking at, but it's clear from the studies, when you look at the diversity and the changes, it's a systems change that's happening in these studies, and that's important. We tend to throw out the baby with the bathwater and focus just on the microbiota, but it's having systemic effects, and I think that's something that this GIBA network, I'm hoping, will address with the funding calls. So I just want to briefly go through some of the things that we use to characterize the microbiota and the microbiome, because not necessarily all of you will be aware of the different methods and the limitations. So we have lots of in vivo systems. It was addressed in some of the questions in the prior talk. Ideally, we want to be working with humans as much as possible, because you cannot recapitulate the complexity of human populations using mouse models. Overwhelmingly, the data available on the gut-brain axis and gut microbiota in general is coming from mouse models, and a lot of the findings from those studies do not translate to humans, because mice have very different anatomy in the intestinal tract. They also have very different microbiota compared with humans. So while they're good for looking at some things, they shouldn't be the only thing that we rely on. And as Alex alluded to, there's a push from the UK government to not use mouse as much. So we're moving towards things like using Drosophila, C. elegans, zebrafish. They're attractive because there's not necessarily the ethical considerations, but also because you can do high throughput screening studies with these organisms, and their genetics are really well understood. So you can do nice complex experiments with these and characterize organs throughout the body, characterize the microbiota, and link everything back into cell function quite easily. Because I know for C. elegans, there are knockouts available for every gene in that organism. So it's easy to be able to do tractable studies to get down to mechanistic underpinnings. The thing to say about all these in vivo studies as well is get all the phenotypic data you can get your hands on. So for human studies, you need everything. Everything's a potential confounder. And for a very long time, we didn't include confounder analysis in human studies, but it's incredibly important and massively changes the outcomes of the interpretation of data, and also in the associations that we're seeing with the microbiota. So for a very long time, TMAO was considered bad, very bad, without any proper explanation. But it turns out that if you're metabolically unhealthy, your renal function's impaired. So you've got much higher circulating levels of TMAO than a metabolically healthy individual. And that TMAO can potentially contribute to renal scarring. It's not that TMAO is bad all the time, because in pescatarian populations, we know it's very good for your health. So again, it's the nuance and subtlety. So the more data you have from a cohort, the more informed your analyses can be, the better your mediation confounder analyses, and the more likely it is that your results are translatable to real world benefits for humans. The other thing as well is, if you're working in animal studies, or if you're doing mouse studies, keep the tissues. So if you're just looking at the transcriptome of the gut, or you're looking at leaky gut and you're looking at the microbiota, that doesn't mean to say that the tissues in other parts of the body are not useful to somebody, and it will massively reduce the amount of animal experimentation that we need to do. Because again, as a systems biologist, it's frustrating to me when I see that 15 people have done essentially the same experiment, but they've all looked at 15 different populations of animals and taken 15 different sets of organs, when one experiment could have covered everything. And again, within the GIBA network, please try and work collaboratively to try and make the most of animal experiments and reduce how many animals we need to use in research. We have lots of in vitro systems available. So anybody who's worked at the University of Reading knows well the smell of the fermentation lab. So I did a lot of batch culture and gut model work during my PhD. So essentially, you can take these systems and inoculate them predominantly with fecal samples, but you can do them with any intestinal sample. They're nice as batch cultures because you can control the pH in the environment. So it means they're really nicely, tightly controlled within an individual experiment, and you get nice reproducible results within an experiment. Once you change a fecal sample and even a donor, you'll get different results, but they're nice to look at fundamental biological processes. So you can look at not just the way that the microbiota changes in terms of function and composition. You can also take the metabolites, you can take the peptides, you can look at the proteome, the transcriptome. So they're a nice closed system, and again, they can be high throughput now because we're not anymore. We're no longer using, or we rarely use the big bioreactors. You can do them in 2 mil cultures and get a lot of information out of those. And the picture at the bottom, the biolog system, essentially we're now at the stage where you can do high throughput screening. So you can get 96 well plates where you can have small inocular, look at responses across either individual donors or the same donor to multiple different insults, or you can take individual species of bacteria and screen them to look at their phenotypes, which is incredibly important. Gut on a chip systems have been alluded to. Yes, in an ideal world, along with organoids, they would be the best thing that we could use. But from experience, from working with clinicians who've been trying to get a range of gastrointestinal organoids, so some esophageal, gastric, pancreatic, liver, it's really difficult to get organoid cultures to succeed. It's got a very low success rate. So while reviewers of grants might like to see organoids, actually it's incredibly difficult to get those systems up and running. So we might still have to rely on the immortalized cell lines for a while longer. Possibly the best system currently available for studying host microbiota interactions is this system from Fafanova et al. It's what Chris Stewart in Newcastle uses, and we have here at NTU as well. It's basically a big anaerobic glove box, but you can have the human cells having oxygen piped into them, so they're under normal physiological conditions. So you can then have the environment of the bacteria in their normal physiological conditions as well. So you can get a better representation of host co-culture and the effects the microbiota is having on cell functions. And I think that kind of system is going to be more beneficial until we're much better at producing organoids across a range of different cell types. As far as characterizing microbiota samples, for a very long time now, well, certainly for the last 15 years, we've been using sequencing. So you start with your fecal sample, usually stored now in something like Zymo DNA RNA shield. The reason that we're storing these samples in this shield is because if you freeze fecal samples at -80 or -20 without any preservative, you will lose important components of the microbiota relatively quickly. Bacteroides, you see a dip in the numbers of those within a month of being stored at -80 or -20. So storing the samples in preservatives is incredibly important. The DNA extraction method as well is incredibly important. You need a bead beating method to get most of the bacterial cells broken. If you're working specifically on fungi or archaea, you may need tailored approaches depending on those organisms. But basically, once you've got your DNA, there's 2 ways that we can look at the microbiota. We can do targeted amplicon sequencing, so that's 16S RNA gene sequencing for bacteria and archaea, or 18S RNA or ITS amplification for fungi. Illumina sequencing, high throughput. Obviously, people are moving towards long read sequencing, but that's too expensive at the moment to be and too labor intensive at the moment to be tractable across lots and lots of research labs. So Illumina sequencing is here to stay for a while. The other option is shotgun metagenomic sequencing. So you take your entire fecal sample, you sequence all the DNA in it, you get your human, bacterial, viral, and fungal DNA, and then you have to make sense of what's going on. Now, I'm talking about feces here initially, but if you're working, well, across all studies, whether you're working with low biomass samples or high biomass samples, such as feces, you should be including community standards in your sequencing runs because it gives you an idea of how successful those runs have been and how efficient the sequencing has been. And it's incredibly important for the 16S based approaches. Shotgun metagenomics is a bit more forgiving, but 16S, every stage from sample collection, extraction, PCR onwards will affect your PCR results. So it's incredibly important that you have this microbial standard in your samples. And similarly, if you're working with low biomass samples, you really need to understand ketone contaminants. So your PCR reagents have contaminants in them and they will influence your results. And there's some really lovely papers from Zana Salter that I thoroughly recommend you go and look at if you're going to be doing any kind of low biomass work. Once you've got your 16S data, I have no preference over whether you use Mother, Dada, or Chime. It's all down to personal preference and which one you find the most intuitive to use. But essentially, they all do the same thing. They take your raw data, merge your reads, get rid of any any low quality sequencing reads, there are processes available across all these 3 packages to get rid of chimeras. So all the chimeras you've got is the 16S gene from one organism has joined another one. So you've basically got what looks like a novel species, but half of it can be Bacteroides and half of it can be Clostridia. It's just noise you need to get rid of. They've all now got packages within them to get rid of contaminants. But if you've got those negative controls in place that you should have anyway, you can also remove the contaminants that way. Because if you're picking up reads in just DNA kits with no bacterial sample in there at all, those are contaminants associated with your DNA extraction kit or your PCR kit, so they can be removed. All of these pipelines give you measures of alpha diversity, so that's basically within a sample, your things like your Shannon-Weaver diversity. It annotates your OTUs, or if you're looking at DARDO2, we call them amplicon sequence variants, but basically it's the same. They're the same thing. It's how you cluster your organisms based on a defined threshold, sequence similarity. And then you've got your beta diversity, which is looking across samples, so things like Bray Curtis. And then there's lots of packages available. These are the ones that we've used, but there's endless lists of packages available for processing those data. So you can either look at them in terms of abundance data, or if you're using ALDEX2 or DSeq2, you use count data. Shotgun metagenomics, there's lots and lots again that you can do with these. There's lots of different packages available. If you're working with human samples, especially complying with GDPR, and you're depositing data with European, EMBL, sorry, in the European Nucleotide Archive, you absolutely need to get rid of the human DNA in samples and discard that unless your ethics approves you to use it, because lots and lots of old shotgun metagenomic data available publicly has lots of human DNA in there, and that has been used to assemble human genomes, which you might not think is a problem, but if you're taking samples from poorly sampled nations, low middle income countries, there's a potential for exploitation of human genomic data. So you need to remove those human reads and just bin them and just focus just on the microbiota within those samples. There's lots of ways of looking at the taxonomic abundance across samples. Metaflan is a marker-based method. It's not great in terms of you're not able to change the database, which is why I don't tend to use that so much. With Kraken 2 and Self, you can build your own databases, and that's incredibly useful if you're looking at lowly sampled sites in the body and you want to get an idea of the proper diversity there. You can do lots of functional analyses, so basically mapping the reads or the genes present in your samples onto known databases of functional pathways. So things like KEGG, which has pathways for every organism, and that's essentially where that rat figure I showed you earlier came from. You can do community level analyses. So obviously there's lots and lots being done on metagenome assembled genomes, and that's massively increased what we know about the diversity of the microbiota and improved the tools we've got for profiling the microbiota. But then obviously you've got things like phyloFlan to look at the phylogenetic analyses, and Metaflan allows you to do species level analyses, potentially at a strain level, but that comes with caveats, which I'm not going to go through here. But strain level analyses or species level analyses are still really difficult to do with metagenomic data. The other thing that you can do as well with the metagenomic data is you can now be in a position to be able to predict the microbial biomass. And this is a nice study that Sean Gibbons' group did, where they looked at flow cytometry, qPCR, because it's much better if you can normalize your metagenomic data back to actual bacterial cell counts. They've shown that you can use the bacterial to host read ratio. You can use that as a proxy for predicting microbial biomass. And we've done this recently with cancer patients, where we've shown that even after resolution of cancer, so these were people who had cancer, have been treated successfully and are now healthy 6 months after their operations. If you can compare them with healthy individuals, they have a far lower biomass potentially than healthy individuals. And that's something to bear in mind, is that your diseases, it might not just be that the microbiota function is changing, the composition is changing, the biomass may also be changing, and that will have knock-on effects in terms of how you interpret data. I will mention culturomics because I think it's incredibly important. And as I've said earlier, we can only annotate up to 30% of genes in a metagenome. sometimes it's down as low as 10%, and that's the bacterial genes. Don't even think about the viral genes. That's even more of a mess. There's a lot to be said for going back and doing culturomics work or microbiology across a range of conditions. We know that we know from studies that Tom Clavel's group's done and Trevor Lawley's group's done, there's huge diversity within samples. We've probably cultivated about 75% of the human microbiota. There's still a lot to be cultivated. And depending on what you want to do, you can just do global screens to just try and isolate everything, or you can do function-driven screens where you know there's a specific bacterial function contributing to an output you're seeing in the gut-brain axis. So you can look for the organisms that can carry out that function, or you can just enrich. So if you're doing things where you want specifically bacteria that are really good at breaking down dietary fibers because you want improved prebiotics, those kinds of approaches may be more useful. And we've gone from doing things on plates to being able to do things in high throughput 96-well plates or droplet assays, so down to single cell resolution. So there's lots still to be done, but getting the microorganisms is one thing. We still need to be better at characterizing their function. And again, high throughput methods are coming through where we can do that now in more detail. Going back to low biomass samples as well, there's a lot to be said for plating out those samples and seeing what you can get off the plate and getting an idea of what actually is in those samples rather than relying on sequence-based results only, because sequence-based results only don't tend to give you an idea of what's living and what's dead. So we've done a lot of work recently with esophageal and gastric biopsy samples and just plated them out and then scraped everything off the plates to do metagenomics on. And we've also been able to build a culture collection of bacteria directly relevant to those environments. And they are very different from what you will see in the large intestine. But this is just to highlight that just taking everything we swept off, we could see that there were 21 different genera of bacteria represented on those esophageal or gastric biopsies. So way more diversity than you'd expect to see, even though you're only seeing maybe 1,000 cells in some samples, a million occasionally. They're really low biomass. But we can use bioinformatics tools to not only tell us which bacteria there, but also to predict the quality of the genomes. So the green circle, essentially, if they're light green, they're high quality, so almost complete genomes. Dark green, it's half a genome, but it's still good enough to get information out of. We can also predict the oxygen tolerance, so whether or not they'll survive aerobically or survive without air or anaerobically. And that means that we can target our isolation methods to really pick up the full diversity of the organisms present in samples. There's not much more to come from me, I promise you. We've got metabolomics. So as I mentioned, we've got lots of metabolites that the microbiota produces, either in the gut or systemically. What you pick up in the gut, it bears no relation to what you pick up in blood and urine. So you need to bear that in mind when there's no correlation between what's present intestinally and what's present systemically. And you need to bear that in mind in your studies. You can do untargeted assays where you basically screen for everything. And historically, we've done that with NMR because it's cheap, but there is a relatively high limit of detection with NMR. So you can do mass spec based methods where you can get down to femtomolar, picomolar quantities, and some of the microbial metabolites will have effects at femto and picomolar quantities. I'm thinking of the ones that contribute to antibiotic-associated hemorrhagic colitis. You don't see them in very high levels at all, but their effects can be devastating. You can do targeted assays where there's a specific set of metabolites, so either short-chain fatty acids, for example, methylamines or indoles that you want to look at. You can have a targeted approach to get those. Semi-targeted is sort of a mixture between the two. So the Biocrates plates are a good example of that. But whatever you do with metabolites, make sure you work with people who know what they're doing. And also, and this goes for all of the omics methods as well, before you process your samples, randomize them. You do not want to artificially introduce technical artifacts that make it look like the separation of samples based on group, where actually you run all your healthy first and all your disease later. These methods are incredibly sensitive to these kinds of things. You need to randomize samples. Ideally, what we're trying to do with all of these different omics, whether it's proteomics, transcriptomics, and I include host transcriptome and proteome as well as bacterial proteome and transcriptome, and host genome as well as the bacterial genomes. We're trying to put everything together to come up with ideas of what's going on with the interactions, and then move, from my perspective, move towards precision medicine. But that applies to precision nutrition, precision diagnostics. Whatever you're trying to do, it's an iterative process, and the more data you have, the better it is. As far as human studies go, I said they're really complex. They don't need to be massively powered. This is a study that we did of 58 patients. The only difference with this study and other clinical studies is these were deeply phenotyped patients. We had their metabolites from their bloods, urine, we had the metagenomics, we had hepatic transcriptome, so all of the genes in their liver samples. We had 120 different clinical markers, and that data set was incredibly well curated. And we were able to demonstrate that a microbial metabolite was directly affecting the disease phenotype of fatty liver disease. So phenyl acetate was directly causing accumulation of lipid droplets. So well-designed studies don't have to have huge numbers of humans, but they still need to be well designed. So please do reach out to people who've worked on these cohort studies. If you've got humans samples you've got access to, because it makes a massive difference. And you need to make sure that everything is processed in exactly the same way all the way through. We would have had 130 patients in this study, but the clinicians changed the RNA extraction method halfway through the last batch of samples, which meant we lost all of them. So minor things like that to save money can have huge implications for outcomes of studies. And finally, with all of these microbiota data, people are moving more towards using machine learning. Think carefully about how you apply machine learning. This is a really nice, it's a 5-year-old paper from Pat Schloss's group, but it's a really nice sort of call to arms and warning for how to use machine learning and when, which essentially decide which approach you need to use based on the question you're trying to ask. Don't use black box approaches and trying to implement them in a lab and you've got no experience. It's really important. And the other thing as well is it doesn't matter what machine learning outputs you get, unless you collaborate with people who understand the systems you're working with, you will never get the full story of what's going on. So as I said, I'm a microbiologist and bioinformatician. I cannot interpret all of the liver disease data that we generate or the brain transcriptome data we generate to the level that somebody who works in that field can. So please work collaboratively when you're working across these systems. It makes a massive difference. And I think I'll leave it there because I realize I'm running out of time. And thank you for your patience at the beginning because of my mess up with the PowerPoint.
That's great. Thanks, Leslie. Applause. Okay, so if everyone has specific questions, I mean, I'm happy to keep the room open. I mean, we do have the session ending at 12, but I'm happy to stay a bit longer.
Yeah, I can stay a bit longer if necessary.
Okay, so we can probably do another 5-10 minutes. Okay, thanks, Leslie. That was very, very comprehensive, and I agree with pretty much everything you mentioned. Obviously, I was working in the microbiomes field as well. I wanted to touch upon some of the things that we discussed briefly with John as well. I think there were questions around that, which is also about the sampling approaches. So what is your opinion? So obviously we're inundated with fecal samples. And as you pointed out, we still know very little about what's actually going on around the whole intestinal tract. So what alternatives should we be looking for? So when we're designing a new study and we want to have a more comprehensive understanding of the full gastrointestinal tract, what can we do?
Well, we've been fortunate because we work with clinicians. When they've been doing reversals of stomas from patients, we've been able to get hold of small intestinal samples. So ileal effluent, duodenal and jejunal effluent. Through the cancer research study, we've managed to get hold of esophageal and gastric biopsies. But as John said, you tend to only get those from people when they're sick. Mm-hmm. We need better ways. So the kind of those smart pills, if there's ones that aren't being developed, and there have been a few iterations where they try and capture samples as they go through the intestinal tract, but none of them's really taken off. Yeah. Because I think the problem with them is they're so prohibitively expensive. It's incredibly difficult to get hold of them. So we need to make better use of surgical samples. It doesn't matter if they're not from healthy individuals, I don't think, because a lot of the time you don't actually see that much difference in the microbiota that you pull off these samples if they're on or off tumors. Essentially, it's the same kind of thing. It's the more fundamental processes you can still study using organisms taken from healthy tissue of a cancer patient versus cancer tissue of a cancer patient. We just have to be pragmatic about things. And
yeah. Okay. So we're starting to have a few questions. So first one, I would like to ask what sequencing depth, GB per sample, would you recommend for MAG recovery in complex gut microbiome samples?
oh, that's a $64 million question. I know that Trevor Lawley uses 2 gigs as standard, doesn't he? And we use 6 gigs as standard.
Yeah.
It depends on the system you're looking at. If you're looking at the sweet plate metagenomic type thing, 2 gig is more than enough to get really good coverage of a lot of bacteria. For gut, so fecal, I would say 5 gig. Obviously, people are now going to much greater depths to try and get strain level resolution, but I think strain level resolution is only going to work if you've got long read sequencing and it works well, because there's still a big problem. Even if you look at the taxonomic data at species level, it's not a good place to be. I would be very skeptical of people saying they've got species level resolution.
Another question. So traditionally, adding too much bacteria to a human in vitro model causes cell death, despite the gut being colonized by, I don't know, I'm not going to read this number, trillions of bacteria in vivo. Does infection of in vitro models just simply require modest balanced amounts of bacteria, or do current in vitro models not allow in vivo levels of colonization by their design? And could they be modified to accommodate this?
It's pretty much impossible to inoculate an in vitro system because we always start with a 10-fold or 100-fold dilution of the fecal sample. So even when we used to count bacteria using fluorescence in situ hybridization, the most you'd see would be maybe 10 to the 9, which would translate to the dilution of what you put in there. I think you could probably go higher. That 10 million is across the entire gastrointestinal tract. In terms of feces, you're talking about 10 to the 11 bacteria per gram of feces. So you would never be able to get an in vitro system up and running that would colonize at 10 to the 11, just for practicality, because it gets so thick and gloopy even if you go to 5 times dilution, that you just can't really stir things properly. It's going to be an issue, I think, when it comes to organ on a chip systems. How do you properly inoculate those, and how do you keep the bacteria actually in the environmental conditions they need to be in? And I don't think that's been addressed yet, but that's a really interesting question.
Okay. For gut microbiome studies, do you recommend identifying mechanisms in vitro before validating in vivo, or is it more advisable to start with humanized mouse models to capture the global effect first?
Well, a systems biologist, you either start with a top-down approach, so you start with the organism and then you go down to the cell, or you start with a bottom-up cell to organism. But what tends to happen is you sort of hover between the two. So it's what systems do you have available to you, and what's the most economical use of those systems. So we tend only to go to animal work after we've done the human work or we've done the cell line work, because we try and minimize the number of animals that we're using. And we try and ask very specific questions in the animal models, just because of the constraints on, and I don't necessarily like using animal models anyway, because a lot of the stuff doesn't translate. So unless you're looking at fundamental mechanisms affecting host cells, it doesn't necessarily translate well. But yeah, realistically, you hover between the two, animals and cell lines.
Yeah. Okay, another question. Thank you, Professor, for an insightful session. Are there any changes that occur at the epigenetic level if the gut microbiome is altered? So epigenetics. Do you know anything about that? Have you done any research on that?
We know there is a group in University of Essex who has shown that you get histone modification with butyrate and acetate. So it definitely happens, and we know as well that you get changing of the homing markers in immune cells as well, depending on what you treat them with. But yeah, I can't remember his name, sorry. Niara Barraza is doing a project with them, and it's all around epigenetic and histone modification. So that's definitely something that's going to be a lot more coming out of, because there's a big LMLC grant that's just been funding on that.
Excellent. There's a question more on transferring samples. So what are possible methods for transferring samples from a field area to another country for microbiome analysis? I guess it's more about safe storage and shipping of samples from field work
So
to the lab?
yeah, it used to be an absolute nightmare because in a lot of settings, especially in low and middle income countries, they didn't have access to fridge and freezer space easily. So that's where the Zymo kit's really useful, because that kit is stable for a few days at room temperature. So it gives you time to get it into a fridge, or you can send them through the post. So American Gut originally, when they started sampling, they just had people sending fecal samples through the post, but then they had blooms of Enterobacteriaceae, so it was massively inflating the Enterobacteriaceae counts. But the things like the Zymo kits and other preservatives stopped that happening, and it's made things a lot easier. It's made it a lot more tractable to send things from field setting.
Excellent. Do you have any general advice on the hypothesis generation and data interpretation on systematic biology to avoid ultimately getting lost in the big data?
Systems biology, we're not supposed to have hypotheses. I keep getting told off for putting in grant applications where there's no hypothesis, because they count them as fishing expeditions. There are sufficient data publicly available now across microbiota studies that hypothesis generation isn't such a problem anymore. It's just read around the area and see what other people have done. Don't take it as gospel that the latest big paper in Nature is the full story, because that's part of the problem, is everything gets inflated and then you get dogma, and you need to keep an open mind. But yeah, hypothesis generation isn't such a problem now from a systems biology perspective, because there are so many publicly available data. The big problem with a lot of the studies that's been done is the poor study design initially in some of these. So you can see there's potential technical artifacts rather than biological artifacts. So I would say the study design is more critical than the hypothesis generation now.
Mm-hmm. Yeah. Another one. So thanks for the amazing talk, Professor. Regarding host genetics and microbiota composition, in individuals genetically predisposed to non-communicable diseases like cancer or diabetes, have we identified a distinct microbial or metabolic signature that exists before clinical diagnosis, or is the metabolome indistinguishable from healthy controls until the pathology manifests? So it's basically cause and effect, cause and correlation, right?
So as far as profiling the microbiota is concerned, and this is something I feel strongly about because of the companies exploiting people, there is nothing medically actionable that can be generated from a microbiota sample. So just because you've got a high abundance of bifidobacteria or a low abundance of bifidobacteria, it's meaningless, because you could sample yourself the next day and it will be a different result. So the companies that suggest that they can predict dietary outcomes or disease outcomes from microbiota profiling, it's a no-no. As far as metabolomic data is concerned, the only thing that we can still say unequivocally that is diagnostic is glucose for diabetes. There's a lot still to be done there. And because the uptake of the metabolomic approach has not been as, we've had lots and lots of metabolomic data, but it's not been applied at metabolites. It may get to the stage where we can profile people and say these people are at higher risk of liver disease because they've got high levels of phenyl acetate in their blood or urine. But we're not there yet. It's something I think that is going to be coming through, though, in the not very near future. And I say 5 to 10 years in terms of near future because it's clinical work. But yeah, it's important.
Great. So what do you see as the most pressing priorities for the microbiome research community in the next years in terms of methodology, reproducibility, and translation?
I'd like people to stop sequencing things and do mechanistic work, because we're at the point where the sequencing data aren't telling us anything we don't know already. It's almost like if you've got somebody saying, I've got people with cancer versus healthy, it's like, I can tell you already what the difference is in the microbiota is probably going to be broadly. I think in terms of exploiting differences between individual microbiotas, things like cancer therapies, we know definitely that the composition of the microbiota affects the efficacy of checkpoint immune inhibitors. We also know the microbiota can change the way in which some drugs work, so metformin and statins. Understanding those mechanisms and how we can exploit them or use them to improve host health, I think, is where things are going in the future. I'm still very skeptical of personalized medicine or nutrition on the back of the microbiota, because everybody's so different. It would be too prohibitively expensive, but there are definitely things that can be used to treat diseases that we could exploit the microbiota with. So, and if somebody's missing an organism that could activate a checkpoint inhibitor, you could potentially give them a probiotic with that function to improve their cancer treatment outcomes. Those kinds of things, I think, are going to change things.
Yeah. So do the microorganisms present in the gut help healing the epithelium lining of the gut?
Well, we know that Akkermansia Akkermansia mucinifera degrades the mucus lining, so basically keeps the mucus being produced, and that can help keep the lining healthy. We know that butyrate production in the gut is incredibly important for intestinal function and energy. Don't think there's anything being done specifically on damage in the gut in terms of putting bacteria in there to improve healing, but certainly we know there are specific bacteria or metabolites that are associated with intestine, with the health of the epithelium. That's what I'm trying to say.
Excellent. Do you think the use of cell-free supernatant from fecal samples in vitro models is a reliable approach to distinguish microbiota-mediated effects from the direct effects of toxins?
So you're talking about toxin-producing bacteria or toxic metabolites produced by bacteria, because they're two different things. So you can, looking at supernatants is useful for seeing if there's a stimulatory effect or an inhibitory effect, but ultimately you'd have to go to individual components. So you could use the global supernatant and then fractionate it to try and see if there's differences, and that's been done for things like the pyrazinones that affect histamine signaling, and also for teilovaline and telomycin, which contribute to antibiotic-associated hemorrhagic colitis. But you generally only get toxins produced if you've got toxinogenic strains of E. coli or CLOS in your systems anyway. We don't understand the diversity of true toxins within the microbiota properly in healthy individuals. So yeah, I don't know if I properly answered that question, but I tried.
And then we have another question. So would you recommend conducting a general exploratory analysis on a human stool sample before a targeted approach? Because it relates a bit with the previous question.
So again, it's where, are you talking about microbiota sampling or metabolite sampling here? So for microbiota, if you want to target specific types of bacteria, you would be better off designing methods to specifically target those. So as I said, the archaea and fungi have different perspectives. If you're looking at fecal samples for metabolomics, it's incredibly difficult to do metabolomics on fecal samples because of the complexity of the matrix. So you have to look at the organic and the inorganic phases, and then you've got the lipid and non-lipid phases as well within there. So you would be much better off using a targeted approach for feces. And most people for fecal samples have just looked at short chain fatty acids because of the long known health associations between those metabolites. And that's why you don't tend to see so much literature on fecal metabolites, is because it's so difficult to do it properly, and it's really messy. It's not nice. Yeah, that's why we tend to worry more about blood and urines, because they're so much more accessible, and we know that we've already got developed assays for targeted and untargeted across those. And as John was saying, some of the metabolites you see in blood and urine, you don't see in feces. So bile acid metabolism, are you really getting a true picture of what's going on if you're looking at fecal bile acids when most of them are taken up in the terminal ileum? So it's knowing a bit about the physiology of the system as well.
And we have a last question. Does orlistat cause epigenetic changes of liver?
Oh, my PhD was on orlistat. No. Orlistat doesn't seem to have any, well, not as far as I know anyway, in terms of orlistat and epigenetic changes in the liver. We know that in terms of the microbiota, it doesn't change the composition. And the microbiota microbial lipid metabolism doesn't seem to be really affected by orlistat. And I only say that after screening thousands and thousands of isolates. But you know, that's a really nice question. But I do think that the amount of exposure of lipids that your body gets because of, so for the benefit of those of you who don't know, orlistat basically stops pancreatic lipases working. So you can have up to 200 grams of fat going through your intestinal tract every day. So if you're on it and you drink something like a full fat latte instead of a skinny latte, you can have uncontrollable fecal fat excretion. It's horrific. I don't think it's necessarily in the system long enough to cause long-term damage, but I don't think anyone's looked at this because it's supposed to be non-absorbable and not taking up into the system. I don't think anybody's looked at the liver changes, but that's a really interesting question and something that probably does need looking at now that more people are taking, well, orlistat's been superseded by GLP-1 receptors, but if people are going back to having to manage things using orlistat, the numbers will go up again.
Okay, and we'll take this last question so we don't extend too much. So what do you see as the biggest gap in our understanding of how microorganisms transition from commensals to pathogens within the gut ecosystem?
They don't. They basically, the pathogenic organisms are things like E. coli 157, which you ingest from things like contaminated meat or listeria. The organisms in the gut aren't suddenly the commensals in the gut aren't suddenly deciding to get big teeth and start attacking you. It's changes in the system. They're just responding to them. So it's a bit like in the lab. If you grow them in different media, they'll react in different ways. If you fundamentally change their environment because of the entire system changing, they're going to just respond. So they're not pathogens. Pathogens cause infections. Commensals in the microbiota are just responding to their environment. So they're just doing what we would do. Like if you poke me long enough, I'll go and do something else. I think that it comes back to what I was saying earlier, is there's no things like good or bad bacteria or good or bad metabolites. It's the context. So if you're healthy, your commensals will be doing what they should be doing. If you're unhealthy, your commensals are just responding to whatever environmental pressures they're under. And I think we need to be more nuanced in the way that we look at things.
Excellent. Thanks, Leslie. Okay, let's thank again John and Leslie for these great talks. And thanks everyone for joining. Apologies for taking a bit longer than anticipated, but hopefully you all found that useful. The recordings will be made available to everyone, so you'll be able to look back and also consult if you had any specific questions. I assume Leslie would also be okay to get any further questions by email when people reach out.
Yeah, that's fine. That's fine.
Okay, excellent. And we'll be back then on Wednesday for the brain session with Marta and David. Thank you all for joining.